Now let's come to biosimilar infliximab, which has caused a lot of discussion since it was licensed in the EU. You can easily see that it's a much more complex molecule. It's glycosylated, and the concern is or was the use in inflammatory bowel disease, an indication where the biosimilar infliximab has not been studied. However, when you look at the European Public Assessment Report, it's a very well-written assessment report, I have to say, and it explains very well what CHMP discussed and decided and how CHMP came to their conclusion. And what you can see is that actually the primary, secondary, and tertiary structure were essentially identical. There were comparable post-translational profiles, comparable in vitro functional characteristics, comparable pharmacokinetics, which was shown in a very large one-year clinical trial in patients with ankylosing spondylitis. And this study also provided additional safety and efficacy study. And then a comparable efficacy, safety, and immunogenicity profile was shown in a large one-year trial in patients with rheumatoid arthritis. And here are just some data that are publicly available. So you see very similar serum concentrations of the reference and biosimilar infliximab at steady state. And here the main results from the confirmatory trial in patients with rheumatoid arthritis showing very similar efficacy results for the main endpoint ACR20 at week 30. And I should also say that this primary analysis was supported by the results of the main secondary efficacy endpoints, which are ACR50, 70, DAS28, and so on. And we also saw a very similar safety profile, including immunogenicity. So it was strikingly similar. However, I wouldn't elaborate on this case if there wasn't a problem. And the problem was that the biosimilar showed a lower level of afucosylated species. And why this is important, I would just like to highlight on this sketch of a monoclonal antibody, where you see on the top the antigen binding region and on at the bottom the FC region. And what I would like to highlight is this glycan structure, which can come with a fucose attached, or then it's fucosylated, or without a fucose attached, then it's an afucosylated glycan structure. And this sits right at the core of the FC binding region. And why this is important is it influences the binding to the FC gamma receptor, which can then change ADCC or CDC activity. Having said that, I come back to our biosimilar infliximab, which showed a lower percentage of afucosylated glycoforms, leading to a lower binding to the FC gamma 3A receptor 3A and B, and a lower ADCC activity. So that all was shown in different assays and is very plausible. So there is indication from literature that ADCC may play a role in inflammatory bowel disease, and therefore this finding might have an impact on the possibility to extrapolate to IBD. This was heavily discussed, but then it was found that the difference in ADCC activity was observed only in the most sensitive in vitro test, which was a very unphysiological test, but highly sensitive. And that's what we want the applicants to do. They should use highly sensitive tests. So we are really sure that we see all the differences there are. But that comes at the disadvantage, of course, that you often see differences and then you don't know, do they have an impact on efficacy and safety or not? But this difference was not observed in conditions that were more physiological. And the applicant did a series of additional functional tests to convince us that this difference was really of no clinical impact. So just to mention some of them. So they, for example, added diluted serum or whole blood to this assay, or they used a wider variety of immune cells, so peripheral, not just natural killer cells. And then the difference in ADCC was not observed anymore. They also investigated the role of ADCC in inflammation, and so they used LPS-stimulated monocytes as target cells, and then no ADCC response was seen, suggesting that ADCC is likely limited in inflammation. Also, no difference was observed in the binding to neutrophils, and the neutrophil cells carry high levels of Fc gamma receptor 3b. And in addition, they could show similar blocking of TNF-alpha effects on human epithelial cells. And finally, they showed similar induction of regulatory macrophages, similar inhibition of T-cell proliferation, and similar promotion of wound healing of human colorectal epithelium cells in an in vitro model. And all this evidence, in the end, convinced CHMP to approve all indications of the reference product. And I cite here from the European Public Assessment Report, very shortly, the reasoning in that for this extrapolation, that the neutralization of the soluble TNF-alpha and transmembrane TNF-alpha mediates the effect in RA and other forms of autoimmune arthritis, so the extrapolation to these other forms was not considered a problem. And that by using a range of experimental models that are considered representative of the pathophysiological conditions and mechanism of action of infliximab, convincing evidence has been provided that the observed difference in afucosylated species is not clinically relevant. And with that, I would like to come to my conclusion, that extrapolation of data is not a new concept, and it is based on sound scientific principles. That extrapolation takes into account the totality of the evidence from the comparability exercise. And finally, in case of doubt, we always request additional data. And with this, I would like to thank you for your attention.
Thank you, Dr. Weise.